very happy to have our speaker, Daniel Tamarit. And he is the assistant professor at the Theoretical Biology and Bioinformatics group, specialized in comparative and evolutionary genomics. And Daniel is currently forming the Archaeal and Bacterial Evolution team, which focuses on fundamental questions in genome evolution. So today he's going to talk about his newest research. So we're very happy to have you here. And so now we have all ears to your stories. It's yours, Daniel.
Great. Thank you so much for the introduction. I was really looking forward to the meeting today. So as the introduction was saying, basically, I'm going to be working on all computational things. So getting to know this community is, I think, going to be extremely invaluable for the next year. So very happy to meet you all, and I'm really looking forward to interacting. So yes, I'm going to be presenting many work that I've been doing as a postdoc in the last year. So I was a postdoc in Wageningen University with Thijs Ettema. And there, our main interest was trying to understand the evolutionary origins of cellular complexity. And in order to explain a bit better what I mean by this, perhaps it's good to take a bit of a step back and talk about this thing that we call the tree of life. So the tree of life is just a very useful metaphor that we use to explain the genealogy, the main relationships between lineages of life on Earth. And we can basically just draw the whole diversity of the biosphere and the way that all of these lineages relate to each other. And when we look at it, there are some things that become pretty apparent. So for example, we can see that the organisms that we see with the naked eye, so animals, fungi, plants, they are really confined to a relatively small fraction of this tree. And well, this is a tree that I drew for aesthetical purposes. If I really was faithful to the actual diversity that they represent, this would probably be a much more minute fraction of the tree. But essentially, these organisms and a bunch of additional microbial lineages that are closely related to them, they exhibit pretty complex behaviors. And a great facilitator for all of these behaviors was the origin of their cellular structure, their cellular architecture. So I'm talking about the origin of the eukaryotic cell. So nowadays, we divide all the organisms that we know into these three groups. So there's eukaryotes here in pink, there's archaea, and there's bacteria. And altogether, this is everything that we know about life on Earth. So the origin of the eukaryotic cell was a pretty stark transition that we still don't really understand. And there's been a lot of research about it, and definitely a lot of things that we have gotten to understand about it. But there's so many different aspects that we just have no clue about. So this is exactly what my talk is going to be about today. So some things that we know. By now, it is pretty well established that the origin of the eukaryotic cell was the result of some kind of symbiotic fusion between two different organisms, at least two different organisms. So there was one bacterial partner and one archaeal partner. And together, somehow they fused, and the result, in some way that we still don't understand, became the eukaryotic cell. So when we try to explain how this first eukaryotic cell originated, we still have a lot of work to do. And one of the things that we try to do is to basically pinpoint each one of the aspects that makes a eukaryote and try to understand how it evolved. So there's been definitely a lot of work over the last decades that has shown a lot of progress in this arena. And some things that we can see, I'm going to try to make so at the moment, Zoom is not really letting me to see the whole slide. There you go, this is better. Okay, so some things that we can see are that, for example, mitochondria are the result of basically the inclusion of the bacterial partner. So they are the remnants of the ancestral bacterial partner. And this partner brought along a bunch of genes for multiple processes. Some of these have to do with metabolism or membrane biology. And this is some of these genes are pretty well established that they came from this ancestor. On the other hand, there's a number of very key functions for cellular processing, like replication, transcription, and translation, that is very clear that have a very different ancestry, and particularly they originate from some organism that was related to the archaea. More recently, it has also become obvious that the cytoskeleton, particularly the actin cytoskeleton, seems to originate from a similar kind of ancestry. So these set of genes are more or less well established that they come from some point of the bacterial and archaeal trees. But there's still a lot of different genes, a lot of different functions, and a lot of biology that is very characteristic of the eukaryotic cell that we still don't understand at all. And very notably, there's a very complex system of endomembrane compartments and vesicles that allows for a lot of versatility of eukaryotic cells. And we still don't really understand where this came from. And then, of course, most notably, there's a nucleus in eukaryotic cells that defines the name of the group itself. We still don't know where this came from. So there's a lot of research to try to understand much more of the different specific characteristics of eukaryotes, where they came from, and in which kind of process they were originated. So there's a number of reasons why we know relatively little about this evolutionary transition. And one of them is that we still don't really understand, or until very recently, we didn't really well understand what kind of ancestors these were. So there was one particular set of analysis that tried to establish the relationship of eukaryotes with respect to the other domains of life, and particularly when referring to informational processes. So this had to do with basically understanding the archaeal ancestry of eukaryotes. And for many years, this was the tree that was canon, that basically you would find everywhere. You still find it in textbooks and in a lot of scientific literature, of course. So in this tree, eukaryotes are shown as a separate domain that is a sister group to Archaea, but is separate to it. So this is what we call the three-domain tree of life. And there was a competing tree that was established, well, that was suggested more or less at the same time, that suggested that eukaryotes were more closely related to a specific group of Archaea that at the moment were called eocytes, and then the rest of the Archaea were perhaps more similar to bacteria. And this was suggested to be a two-domain tree of life. So for many years, the three-domain tree of life had been the most popular one. And there were some analysis here and there that showed different kinds of relationships of eukaryotes with some specific groups of Archaea. But this was always very unclear, and definitely there was more data needed. And these data definitely started arriving with the advent of metagenomic analysis. And particularly, there was one study that came out in 2015. So these had to do with marine sediments of a region in the Arctic Ocean called Loki's Castle. So it's called Loki's Castle because it has this interesting rock formations that almost resemble some Asgard mythological scenarios. And in this region, there were a lot of interesting organisms to be looked at, but one of the most interesting ones was a group of Archaea that was called Loki Archaea. And there were some preliminary evolutionary analysis that showed that these Loki Archaea seem to be the closest relatives of eukaryotes. And in the next years, so that was 2015, and there's been, of course, a lot of additional metagenomic analysis that have recovered many more genomes that are related to these Loki Archaea. And collectively, this group has been called Asgard Archaea, and now we know a lot of the sub-lineages of Asgard Archaea that we call Heimdall Archaea, Odin Archaea, Thor Archaea, etc. And collectively, somehow, Asgard Archaea seem to be the closest relatives of eukaryotes. So we still have to really understand what's the exact relationship of Asgard Archaea with eukaryotes, but that is where the current analyses are pointing at. So, so the question is, what is the ancestry of eukaryotes? What is particularly the archaeal ancestry of eukaryotes? And in the last decade, maybe the last 10 years, there's been a lot of advances in metagenomics that really allow us to access much more genomic data that really helps getting to new solutions for this problem. And there's been a lot of computational development for particularly deep evolutionary analysis. So getting to do like complex phylogenomic analysis is something that is more and more possible with the large amount of data and the large amount of, let's say, the sophisticated evolutionary models that we have at the moment. So in this context, in 2016, the group of Thijs Ettema started gathering data from multiple sites around the world and started assembling a team to really analyze all of this. And I'm talking about 2016, but so this is definitely a project that has lasted a very, very long. It was originally coordinated by Thijs himself and Laura M. So Laura has been really invaluable for this project. She is now leading a lab in Paris. I joined in 2017, at the end of 2017. So I've been involved in this project from almost the beginning. And I will explain a little bit more about my specific part of it. And Eva Cáceres was a PhD student with Thijs, and she was really important in the very first steps of the analysis, really getting all the genome data at a very high quality and getting things running from the start. Besides that, there's a number of people that have contributed different kinds of analysis, definitely a lot of data, and it's really been a very nice team effort in that sense. So this paper, I'm going to focus mostly on it. I will also explain a little bit about other projects, but I'm going to present mostly this paper. And this is basically in bioRxiv. You can see a version that is almost final in bioRxiv, but the final version will come in Nature, I think in 10 days. So yeah, so it should be ready for, to see for anybody. So in this paper, we do a lot of different things. Mostly, I think our main contribution has to do with the phylogenomic analysis. And to cut a very, very long story short, we did a number of different things. One of the key parts was that we performed analysis basically twice. So we submitted this paper originally in 2021, and then reviewers were quite positive, but they told us that we needed to include new data that had been published basically months before our submission. No, it was right after our submission, months before the revision came. So then we incorporated all of that data, and fortunately the results were exactly the same. So this really added a lot of weight to the conclusions that we were reaching. Besides that, we also used 2 different gene data sets, and these became also pretty important. I will explain briefly why in a couple of minutes. And we tested the effect of including multiple different groups in our analysis. And this was particularly important in this kind of analysis because long branches in phylogenomics can lead to pretty confusing artifacts, and yeah, so disentangling the errors that can be added by adding long branches from other kinds of solutions is pretty time-consuming. And indeed, Archaea and eukaryotes were some of the groups with the longest branches, and really understanding the effects that they had in our phylogenetic was a lot of work. Besides this, I will explain a bit better the effect of data processing. This was really essential to really get to squeeze out very deep evolutionary signals from our alignments. And we performed also multiple methods to try to see if they converge to the same solutions. And fortunately, they did. So very briefly, I didn't want to explain this in a lot of detail because I'm pretty sure that this is something that is either relatively familiar or maybe not extremely important in the very detailed aspect of it. But phylogenomics is performed nowadays mostly through a method that is called the concatenation method. And in order to analyze trees under this method, what we do is to gather a number of gene markers that are suspected to follow the same evolutionary history. We assess whether that is the case or not. We try to remove all of those gene markers that in principle seem to show signs of some other kinds of events, horizontal transfer, or compositional complicated things that can maybe lead to different topologies. And then if these trees are suspected to indeed evolve under the same evolutionary history, they are taken all together, concatenated, and then we apply relatively realistic evolutionary models on them and perform phylogenomic analysis using basically a single long alignment. So doing this analysis, from the very early beginning, we did see a very clear relationship between eukaryotes and Asgard Archaea, as we were indeed expecting. And particularly, there was a very close relationship with a group of Asgard Archaea that was called Heimdall Archaea. So this is something that was already kind of hinted at in early analysis, and some other people have been observing similar things in the last year. So this is quite reassuring. And on top of that, there was this other group that we called, that is called Njord now, Njord Archaea. And in some of our phylogenies, they seem to be the closest relative of eukaryotes. So this was very interesting. And they have very peculiar biology that made scenarios for eukaryotic origins quite appealing. So these are hyperthermophiles, and potentially a thermophilic origin of eukaryotes could be a very interesting thing to find. But then we had some other cases in which this Njord went completely somewhere else in the tree. And understanding exactly what was happening there took us a lot of work. It turns out that indeed the thermophilic compositions of the genomes of Njord Archaea was similar to other thermophilic adaptations happening in other places in the archaeal tree, particularly in core archaea, who are also thermophilic. And then these kind of relationship was artifactual. So basically, Njord were pulled out of Asgard Archaea because of their thermophilic compositions. And indeed, we can see that when we analyze the sites of these alignments that are less prone to these thermophilic adaptations, Njord go back inside the Asgard archaeal tree. And we have additional evidence, like in terms of the gene content of this archaea, that also show that they are clearly Asgard archaea. So that was indeed a very conclusion to have. Now we know that Njord are Asgard archaea, they are somehow related to Heimdall, and we really wanted to understand what was happening in this particular side of the tree. So maybe just to reiterate, there is this particular group here that we call collectively Heimdall archaea. The taxonomy of Asgard archaea is in flux and is pretty complicated, but at the moment, maybe we don't really need to specify more names. So these are called Heimdall archaea, and these include this Njord archaea. Now we are pretty sure that they are somehow inside the Heimdall archaea. And then there's this other subgroup of Heimdall archaea that we call Hod archaea. And somehow we know that eukaryotes are related to this group. So we did a number of analyses that really tried to specify where exactly in relation to Heimdall archaea were placed, the eukaryotes were placed. So one of the results was the one that I just showed. Eukaryotes were sister to Njord when the alignments were used basically as we obtained them from the alignment algorithms. Then we also performed a number of data processing analysis to really try to squeeze out very, let's say, deep evolutionary signals. So one of these things that is relatively common to do is fast site removal. So this is one type of data processing by which basically we identify sites of the alignment that are evolving under different rates. So for example, here we can see that there's this particular site that seems to be evolving pretty fast. So there's many different kind of amino acids shown in different taxa, and this is inferred to have the highest rate of this whole alignment. So then we basically do stepwise removal by which we remove, let's say, 10% of the fastest sites or 20% of the fastest sites and so on. And then we try to see what's the effect of doing that kind of data removal. And when we did this, it became pretty obvious that eukaryotes stopped being so closely associated with Njord, and instead they seem to be associated in general with Heimdall archaea, which was a potential solution that we already expected from previous analysis. So it really seemed that the association with Njord archaea had to do with this very rapid evolution that Njord archaea had suffered in order to adapt to basically thermophilic environments. And this long branch was causing this known artifact that is called long branch attraction. So that was already something quite interesting that we could see that was not obvious from the start, let's say. Another thing that we tried was data recoding. So this is a type of data processing by which you identify which amino acids are more likely to be exchanged to each other. So basically they are categorized in subgroups of amino acids that have a lot of frequent exchanges. And then you just transform this into basically one single category. So basically going from a 20-letter alphabet to a 4-letter alphabet. And then you recode the original alignment using this 4-letter alphabet. So by doing this, we can just focus on exchanges that happen in between amino acids that normally do not exchange between each other. So they are slower, more infrequent kind of mutations, kind of exchanges. And by doing this, we can, in principle, remove some of the saturation that exists in these alignments. So when we did this, we observed a similar thing for some of the analysis. So maximum likelihood trees showed that eukaryotes might be related to the whole of Heimdall archaea, basically altogether. But then Bayesian and Inference seem to point a specific other location inside the Heimdall archaea. So eukaryotes here were more closely related to this group that we call Hod archaea. So that was quite interesting. Hod are very different to Njord. They have very large genomes. They are mesophiles, and they indeed have a very interesting set of genes for complex behaviors that maybe I will explain in a bit more detail very soon. Then the other thing that we did was basically to put together this two different analysis, fast site removal and recoding, and then both Bayesian inference and maximum likelihood pointed to the same solution. So eukaryotes potentially were the closest, so Hod archaea were the closest relatives of eukaryotes. So this is indeed not the final, let's say, nail in the coffin. We do believe that this is the most likely solution for the phylogenomic placement of eukaryotes in the Asgard archaeal tree. So Hod might be the closest archaeal sister of eukaryotes. But indeed, we are doing more analysis to try to corroborate this. So one of the things that we can also see basically in parallel to this phylogenomic analysis is the fact that when we look at gene content, if there is one particular Asgard archaeal group that has more, let's say, eukaryotic-like genes in common with eukaryotes, this is indeed the Hod archaea. So I don't really want to go into details here. This is a very complex figure, but basically hot archaea have a very interesting gene repertoire for genes for information processing, cytoskeleton, trafficking machinery, endosomal sorting. So a number of things that are typical of eukaryotes. And many Asgard archaea seem to have a set of these genes, but if there's one group that has a few more in common with them, is the hot archaea. Another thing that we did in order to really try to understand which of these genes basically were inherited to the eukaryotic ancestor is this method that we call gene tree, species tree reconciliation. We as a community, it's not that we are really developing the method itself. But by this method, basically you provide a species tree, such as the one that we obtained in the last analysis, and then you provide phylogenies for each one of the gene families in these genomes. So basically here we had tens of thousands of gene trees. And then we just reconcile these two by basically placing the gene trees inside the species tree. So doing something basically kind of like this. So this method basically recovers gene duplications, horizontal gene transfers, gene losses. And for each one of the nodes in the tree, recovers ancestral gene content. So this was a way not only to really identify what was present at specific points of this tree, but in general to understand general trends of the evolution of Asgard archaea. So there's a number of things that we could see using these methods, but maybe just not to make this story very long. One of the things that we can see in the last archaeal eukaryotic ancestor is that it already had a pretty large genome, definitely very large for an archaeon. And interestingly, it had a pretty high duplication to loss ratio, something that is not really very observed in general in archaea. So this part of the tree, or this part of the Asgard archaeal tree, was in relatively rapid expansion through evolutionary mode that is not very common in archaea, and is a bit more common in eukaryotes, perhaps. In particular, this part of the tree also seemed to have a very interesting gene repertoire for a number of functions, particularly protein targeting was one of the functions that we found particularly interesting. So this is a set of genes that in eukaryotes has basically the role of placing proteins in specific compartments or specific regions of the eukaryotic cell. And we find several of these genes in Asgard archaea. So we don't really know if they have compartments, but at least they may have some kind of cell regionalization. So some proteins go into specific areas of the specific regions of the cell. So definitely there's a lot of work to be done to really show this, but that's definitely very interesting. We also find a number of genes for membrane trafficking and vesicular formation, some of the functions that are pretty essential for the versatility of eukaryotic cells. I will mention a little bit more about this very soon. And additional genes for genome maintenance, gene expression, etc. So some of these were genes that were only found in eukaryotes before, and now they are also found in Asgard archaea, and particularly they seem to be present in this Heimdall archaeal group. Besides this, given that now we think that this hot archaea is the closest the closest archaeal relative of eukaryotes, we also believe that the ancestor of eukaryotes was mesophilic. This also doesn't require very big changes in the eukaryote genetic theory to really accommodate a thermophilic origin of eukaryotes. That would have been definitely a bit of a departure. So we do believe that it was a mesophilic ancestor in that sense. Okay. In parallel to all of this, I was very lucky to be part of a Wellcome Trust consortium together with a number of groups in the UK. And they were really focused on let's say, cellular biology and in vitro studies of Asgard archaeal proteins. And so I was basically just providing them with the bioinformatic analysis that they needed. And this was a very interesting collaboration. And particularly, they were focusing on the ESCRT system that is essential in eukaryotes to basically define membrane traffic and membrane dynamics. And some things that we saw is that indeed these genes are relatively well conserved. So they are a little bit patchy in Asgard archaea, but they are relatively well conserved. And very often they form, they go together in their genomes, let's say, which already suggested that probably they were somehow acting together. And this group of people made a really nice work to show that most of these genes seem to interact between each other in ways that are pretty similar as how they interact in eukaryotes. So for example, this figure puts together a number of assays that come from GIS2 hybrid analysis. And indeed, the image that comes out of this analysis is very similar to what you would see in eukaryotes. So potentially, these genes have similar functions in Asgard archaea. which is very exciting. And there's definitely more work to be done in this side of things, but that's at least a very good start. There's definitely a lot of progress in understanding the cellular biology of Asgard-Archaea, even if there's still a lot of that we don't know. But perhaps something that is worth mentioning is this particular study from Burak Afci. Thijs was also part of it, and additional collaborators. So they did microscopy on a number of Asgard-Archaea cells, and they found this very intriguing gap that exists between the DNA stain here, DAPI in blue, and the ribosomal stain here in red and green. So this is a 16S probe. And so there seems to be some kind of gap, some kind of area of the cell that is not stained by either DNA or 16S ribosomal RNA. And this can be seen from multiple pictures. So whether this means that there's an actual separation through a membrane or something else is, of course, very unclear. The authors were very, very careful not to use the word nucleus in their paper, but there may be some interesting biology happening there. Besides that, the last couple of years have been very interesting also in terms of getting to grow some of these Asgard-Archaea. So they are notoriously difficult to culture in the lab, and there's now a couple of studies that have managed to culture at least one type of Loki-Archaea. And they see this interesting protrusions, these interesting tubes that come out of the cells, and it's very unclear what they do, but they do resemble some of the potential dynamism that exists in membrane biology in eukaryotes. And particularly this this latter analysis showed that there's a lot of actin filaments that might be key to determine the dynamics of these protrusions. And there's additional studies that have shown that proteins that interact with this actin cytoskeleton also seem to be conserved in Asgard-Archaea, and they seem to be performing very similar functions to what they do in eukaryotes. So at least now I think there's a very clear picture emerging that the actin cytoskeleton in Asgard-Archaea is very similar to the one that we can find in eukaryotes. There's a lot still that we don't really understand about Asgard-Archaea, about their biology. Some of the things that we are trying to do in this line to get to understand a bit better is, of course, on one hand, corroborate our phylogenomic analysis. So we still have a number of things running with more sophisticated evolutionary models or more complicated phylogenomic analysis that we really hope will either corroborate the results we have or suggest something very robust that we can really move forward with and then really understand what's exactly the relationship between eukaryotes and Asgard-Archaea. And additionally, of course, we are interested in the cell biology of these organisms and trying to understand, for a lot of very interesting protein families, where did the eukaryotic protein families come from? What is the role of these protein families in Asgard-Archaea? And we do have a couple of things going that hopefully we will manage to publish in the next year or so. So yeah, well, stay tuned for that. So altogether, I think there's this interesting picture emerging that the Asgard-Archaeal ancestor of eukaryotes did have some kind of complexity, some kind of complex behavior, potentially through interesting membrane dynamics, potentially a separation between DNA and the ribosomes, definitely some interesting either cell regionalization or membrane dynamics that does resemble a little bit what eukaryotes can do. And hopefully the next years are going to be really key in understanding more about this biology. Another side of things that I will only present very briefly. So we wanted to get to know more about the ecology of Asgard-Archaea, let's say. And for this, so this is again a long story that I'm cutting a lot, but we started from the perspective of trying to increase the quality of the Asgard-Archaeal genome. So much of the genomes that we obtained through metagenomics were very, very fractionated. So we really wanted to focus on some that we could put all together, close them, and really get to study some of the new things that we could get that way. So we were actually quite fortunate in managing to close one of these genomes. So this was an Odin Archaeon. And here I was also working with Eva Cáceres that I introduced before, and a number of people that really did a definitely very key contribution to this paper. So together, this genome was very interesting because it did provide some aspects that we were completely missing from previous Asgard-Archaeal genomes. And particularly, this was like two very long CRISPR cassettes. So CRISPR repeats, they are interspersed by these CRISPR spacers that are incorporated from mobile elements that infect the cell. So by incorporating these mobile element, small regions, we could understand a little bit more about the mobilome of Asgard Archaea. So this is something that we were after for quite a while. And by doing this, so here I'm just showing basically lines between this CRISPR cassettes and these two particular elements. The different colors just mean different methods. But there's a very clear relation between these two contexts and the spacers that we find in this CRISPR spacers. So these two basically, now we know that they are two complete viral genomes. So basically this is a circular context, and this contains inverted repeats at the end that also suggest that it's a complete context. We call this Hugging and Moonin. These are basically the names of the ravens of Odin, which indeed in the story of Odin, the ravens go around the world and bring information back to Odin. In this case, the viruses probably do a very similar kind of function here. They infect the cell and potentially bring some additional genes into the Odin genome. But indeed, these are two relatively small contexts, typically archaeal viruses. And there's very little that these viruses seem to resemble, let's say, eukaryotic viruses or viruses that we can find in bacteria. So these seem to be very typical archaeal viruses. And in parallel, there's been a number of additional studies that, well, two of them were published basically at the same time as ours. There was an additional study in Nature Microbiology that did basically a very similar kind of approach, by which by identifying spacers in CRISPR arrays, people could identify different mobile elements infecting different groups of Asgard Archaea. And the picture was indeed the same for all of our studies. All of the things that we can identify are either relatively unknown or relatively typical archaeal viruses. So there's, at the moment, nothing yet that suggests that the Asgard Archaea mobilome is anything that resembles eukaryotic mobilome, or at least from the viral side. So this is as much as we know for now, even if there's, of course, a lot of additional projects ongoing, and I think there's going to be some surprises about this coming from at least a couple of groups that I've heard about. But this is the picture that we have at the moment, and well, maybe I will leave it there for now. So yes, maybe just putting things together. Most of the work that I've described today has to do with phylogenomic investigations, and particularly really trying to pinpoint the position of eukaryotes inside the Asgard archaeal tree has been extremely tedious, and identifying artifacts that were really like troubling us for years was a lot of work, but definitely worth it. And we could really pinpoint at least one specific solution that we're favoring at the moment, which is that these hot archaeales or hot archaea seem to be the closest archaeal relatives of eukaryotes as we know at the moment. And by using this species tree, we can also identify a number of properties for this last Asgard archaeal ancestor of eukaryotes. So on one hand, we believe that it was a mesophilic heterotroph, and it had a relatively large genome for an archaeon, particularly increased through gene duplication and decreased gene loss rates. And this ancestor of eukaryotes seemed to have a relatively rich gene repertoire for a number of complex eukaryotic pathways, and particularly some genes involved in protein targeting or membrane trafficking are very interesting. And I think there's going to be a lot of interesting work coming in the next years to show what exactly they are doing in Asgard archaea and how the eukaryotic homologs acquire their functions, let's say. So besides this, we still believe that closing genomes of uncultivated archaea is valuable, even if there's not a lot of emphasis put on this, but it's also becoming much easier through long-read metagenomics. And getting these complete genomes is indeed bringing a lot of interesting information still. At least from the few analyses that have been published so far, it seems that the Asgard archaeal viruses are typically archaeal. They don't seem to suggest a lot in terms of interesting eukaryogenetic scenarios. But yes, indeed, as I said, there might be some other information coming. And I will leave it here for now. I'm looking forward to chatting about this. If you have questions, feel free to ask. And I, of course, will be around. So I'm very happy to stay in contact by any other means. So thank you, everybody.
Yeah, thank you very much, Daniel. Thank you. Very nice talk. And any questions? You can raise your hands or just unmute yourself and directly ask Daniel. See, yeah, this is a question from a friendly Zoomer.
Mm-hmm. Yeah, sorry about my friendly Zoomer thing. This is Jan Kees. I'm still. There's still this other theory that you have this bacteria that sort of eats or is incorporated by an Asgard, and then it's sort of that Asgard becomes the nucleus. And I was wondering if you look at this electronic microscope picture of this long tube kind of things, if this whole thing is sort of incorporated, then you also have sort of a membrane system inside the bacterium. That's archaeal, of course, and also the nucleus is archaeal. So you still get the same phylogenetic trees. And yeah, for me, it can be either way. It's not that it can still be that sort of like a bacterium was the sort of the starting thing instead of the archaean.
Yeah, no, I totally see your point, and I think we already had a bit of a brief conversation about this. I don't really have a strong opinion on the current, let's say, cellular topology models for the origin of eukaryotes. I think there's different ways to express the potential relatedness of the genes that we find nowadays in eukaryotes in terms of what exactly their partners were doing. And I think what is really going to potentially close the debate is really showing what is happening here. I think if this is really shown to be a nucleus, that's definitely going to change the picture. If that's not the case, yeah. So this debate may take a bit longer to solve otherwise.
Yeah. Okay, thanks.
Yeah.
Thank you. Any more questions? I think I think Brian.
Thank you. Yeah, thank you for the nice talk today. I had a question about the condensation of amino acids to 4 classes. Why did you choose 4 classes and why not 5 or 6?
Yeah, that's
do any experiments changing the number of classes? And if so, I'm mainly interested in the loss of information content that you experienced by doing that.
Yeah, that is a very good question. And we are also experimenting a bit with different number of categories. The main reason to use 4 was simply due to the fact that other people had used it before, and they had seen that indeed, as long as you have relatively long alignments, the loss of signal is not really that important. There are some studies that suggest that this kind of categorization in only 4 categories does remove a lot of important information, but everything that I've seen that has been published so far basically includes alignments that are 1,000 amino acids or less. And here we had 10,000 to 15,000 to work with. So indeed, we find phylogenies that are well supported. Normally, if you just don't have enough support, if you don't have enough signal, you just don't have enough support. So as long as we obtain supported topologies, we don't believe that we're losing important information. But that being said, as I said, we're really experimenting with additional kind of categories, and we don't really see differences in the kind of results that we obtain.
Very interesting. Interesting. I think I'll apply something like this myself. Thank you.
I'm very happy to chat about this if you have other thoughts, and yeah, just feel free to reach out if you want to talk more.
Yeah, thank you. And any more questions? Yeah, Berends, please.
Yeah, so thanks for a nice talk. I was wondering about this metagenomics, having these full-length genomes, and of course, very nice that you find these beautiful viruses. But so in terms of ecology, and then still looping back into eukaryogenesis, I guess one of the interesting things is all these syntrophic relations. And do you have any hope of if you integrate enough metagenomic data that you could say something about, if there's any ecological relations between taxa and metagenomic data? I don't know if you need full-length genomes for that, but I mean, if you start mapping back reads, that maybe something pops up.
Yeah, the only thing that I know that people have tried, we also tried, and basically there's a range of analysis that people have been doing with just correlating abundances and trying to see if basically higher presence of specific Asgard archaeal groups may correlate with specific abundance of other microbial lineages. And through these kind of studies, there's not a lot of very clear results, but whenever there's some kind of correlation, they seem to point to similar relationships to those that have been described so far. So for example, I think I did a bit of a long and complicated sentence, but there's a number of studies that have managed to enrich in Asgard Archaea, particularly the Imachi analysis. So they saw some kind of, well, an important relation with a couple of groups that, so basically they could never have these loci archaea in isolation. They were always co-isolated with a couple of other groups. And these may have like actual very, let's say, indispensable relationships. Like you may not be able to grow one without the others. The kind of correlation analysis that I was mentioning seem to show similar things. So similar kinds of groups forming syntrophic relationships with at least particularly some loci archaea, and I'm not really sure if they also found with some Heimdall archaea.
But especially if you now say that these, what is it, Hot Archaeota or Hot Archaeales are the closest, then for those, I think it would be most interesting to find.
Yes, so for those, as far as I know, there's no correlations that have shown anything at all. There's no like conclusive results that I remember at the moment.
But do you trust these methods? Because
right, that's indeed the question. Normally these methods just don't really seem to provide a lot of results. And I guess what people are really trying to do is enrich. So when there's proper enrichments of hot archaea, we will have to see if they manage to grow by themselves or if they need some partners. So far, nobody has really managed to do that.
I think Anya has this, at least for deep end archaea, this notion that transfers are actually a hint of symbiosis.
Right.
Have you seen something like that?
No, I haven't. Something that we are trying to do is in general see what's the extent of horizontal transfers in general in Asgard archaea and see if there's any kind of enrichment in that. But to be honest, we haven't really seen any specific patterns that are very clear yet. Yeah.
Thanks.
Yeah, thanks.
Okay, thank you. Any more questions? Yeah, Carlos, please unmute yourself. Yeah, thanks.
Hello. Yeah, so thank you very much, Daniel, for the talk. I was curious about the distribution of the score system in bacteria. Is there a score system in bacteria?
There are some homologs indeed. So particularly, I know that for the ESCRT-3, there's been relatively recent finding that there's a what's the name, VSP VIP. I'm forgetting the name. But there's a particular homolog in bacteria for ESCRT-3. Not really. Sorry?
The entire complex or the entire operon is not there. It's not in bacteria, right?
Not that I know of, no. As far as I know, so this is the one that this is the one that I remember that has been found in bacteria. For the rest, I don't know. I don't think so, but I'm not really sure actually.
Yeah, because I was curious about the fact that the archaea are supposed to have a very different membrane composition from eukaryotes and bacteria. And this score system is working in G1P or in archaeal membranes. So, and you found actually a patchy distribution, which I was wondering if this patchy distribution could correlate with other different distribution of genes involved in the membranes composition, or for example, if these subunits of archaea of Asgard can complement eukaryotic subunits in other organisms, or to understand this transition of the score system, also integrating the membrane transition.
Yeah. No, I see what you mean. So I don't know about the specific presence in bacteria. I don't think there's been a lot of work on that, but definitely these ESCRT systems are the same ESCRT proteins that work in Asgard archaea. They reconstitute the system in yeast, for example. So they seem to work well with the membrane biology that exists in yeast. So yeah, it doesn't seem to be specific of a particular lipid composition in that sense.
Okay, that's good to know. Thank you.
Thanks.
Thank you. Any more questions? Yeah, and I think that's the end of the talk, and that's the